So welcome back to our biology series. That last time we talked about carbon and water. Now we're going to be talking about macromolecules, basically molecules made out of carbon, large molecules. Like I said, it's a large biological molecule, and most of them are made out of carbon. A monomer is a building block of the macromolecules. So think about a ladder. First, you would need one piece of wood. That's the monomer. The polymer is made out of identical monomers. So the ladder is made out of identical rungs made out of wood. So the monomer is the wood rungs, and the polymer is the total ladder. So basic thing about macromolecules, the dehydration reaction is something most macromolecules go through, and is the synthesis of polymers. This is called dehydration. It's not because it's thirsty, it's because it releases water. So when you put monomers together to create a polymer, this is called a dehydration reaction, and it releases water. The reason it releases water is because one of the monomers provides a hydroxyl and the other provides a hydrogen. We talked about hydroxyl before, and that's basically OH minus. Hydrogen would be H plus. So when these combine, there's an extra H2O. H plus plus OH minus, let me write that down, H plus plus OH minus gives you a water molecule, which is then released. Hydrolysis is the opposite to this, and it's the breakdown of polymers. This uses up one H2O. One monomer receives the hydroxyl and the other receives the hydrogen, and they split apart. Goodbye. So some of the main macromolecules we'll talk about today: carbohydrates, proteins, nucleic acids, and lipids. But lipids aren't really considered macromolecules because they're not large enough. But we'll talk about them anyway. You've probably heard of most of these: carbohydrates, proteins, yes, nucleic acids, maybe, lipids, it's just another fancy name for fat. So first, carbohydrates. A carbohydrate provides energy and building material. Those are the main functions. It contains a carbonyl group and a hydroxyl group, which is why it's carbohydrate. The 8 is just there. So the monomer of a carbohydrate is called a monosaccharide. It's not saccharide, it's saccharide. And it's a single sugar, usually multiple of CH2O. So a probably famous one is glucose, which we'll talk about later. It's pretty important: photosynthesis, cellular respiration, later lessons. And this is C6H12O6. And you can see, if you multiply this by 6, you'll get 6 C's, you'll get 12 H, and you'll get 6 O's. Perfect. It fits the rule. A triose is a 3-carbon sugar. A pentose is a 5-carbon sugar. By the way, this is glucose. A glucose is a 5-carbon sugar or a pentose. A hexose is a 6-carbon sugar. Most monosaccharides are major nutrients for cells. A disaccharide is 2 monosaccharides bonded through glycosidic linkage. So don't worry if you don't know that. It's basically the bonding of monosaccharides to form disaccharides. Basically the opposite of this definition. A disaccharide, as the name suggests, is made out of 2 monosaccharides. A polysaccharide is a polymer of monosaccharides. So an example of a disaccharide would be sucrose or table sugar. You've probably heard of that. And this is basically glucose plus fructose. And fructose is another monosaccharide. And this is similar to glucose. It's an isomer to glucose, by the way. Now, fructose is the same formula as glucose, but different structure. So like we talked about last time, it's an isomer of glucose. Fructose is the sugar mainly found in fruits, which is why it's fructose. And you've probably heard of it before, even if you didn't know the name. A polysaccharide, like I said, polymer. So an example would be starch. Starch is major, long chains of monosaccharides, maybe thousands in it. Yes, starch. It read my mind. Glucose polysaccharide in plants. And this mainly stores energy. That's the main job of starch. Glycogen is the glucose polysaccharide in animals. And this stores energy, but limitedly. Most energy storage is in lipids in animals. Cellulose is a glucose polysaccharide in plants. And this is similar to starch, except it's not starch. It does not store energy. It makes up cell walls. So don't confuse it with starch. Cellulose, starch, completely different, but they both are glucose polysaccharides in plants. Chitin is a glucose polysaccharide in arthropods. Arthropods are basically like spiders, etc. And you know what an exoskeleton is? Well, it's basically that hard shell on top of a spider. If you accidentally stepped on one, you would see that it's like a cracked shell. Okay, maybe that's too gruesome, but still. It's the outside shell of an arthropod, and it's made out of chitin. When I say nitrogen appendage, it basically means it has extra nitrogen. Lipids are the next fake macromolecules we'll talk about. A lipid is a hydrophobic molecule. We talked about hydrophobic and hydrophilic last time. Hydrophobic hates water, hydrophilic loves water. Fat is one of the main types of lipids. I told you that lipids are basically fat. They're not. They're actually, it's more like fat is a type of lipid than lipid is a type of fat. It's like the square rectangle situation. So fat is one of the types of lipids, and it's the main one, but there are other types of lipids that we'll go into today. So fat, it stores long-term energy / food and insulation. So fat basically is energy storage. If you want energy, you burn fat. So basically when you're running, that's why you lose weight when you're running. If you run a lot, actually. If you run like one lap every week, then you're probably not going to lose fat. It probably would use other energy sources than fat. So how to create a fat? Well, you basically add some glycerol and fatty acids through ester linkages. So we talked about glycolipid linkages. Now we're talking about ester linkages. So fatty acids are long chains of carbons, not as long as carbohydrates though, mostly polysaccharides. And when you add it to a glycerol, you get fat. So this is the glycerol. It's normally depicted with triangle. It's not actually a triangle shape. And this is a fatty acid. You see, that's one, that's another, and there's a third. So there's a reason why I drew three fatty acids, and that's because a most common fat is triglycerol. And this has 3 fatty acids, like I drew in the diagram. So you've probably heard of saturated fat before on the nutrition labels, and you probably don't know the difference between saturated, unsaturated, and there's also something called trans, which is completely horrible for you. So first, saturated. The scientific definition of saturated fat is it has no double bonds between carbon. That's probably not the definition you'll see on a nutritional label or in health class. And these are usually solid. So when it says saturated, it basically means that there are no double bonds. None of that. All the bonds between carbons are single. Unsaturated has 1 or more cis double bonds between carbon. So we talked about cis last time, and it has this, at least 1. If it has 1, then it's called a monounsaturated fat. If it has 2 or more, it's called polyunsaturated fat. Those mono and polys, they really form a pattern. Mono just means 1, poly means many. Trans fat is 1 or more trans double bond between carbon, and this is unhealthy. Avoid it. And it basically creates a kink in the fatty acid. So that's bad for your health. It's hard to break down, etc. Just avoid it. Phospholipids. So phospholipids make up the cell membrane of a cell. And we'll talk about cells in our next lesson and cell membranes in the lesson after that. But first thing to note, phospholipids are the things that make up the things surrounding the cells. Okay, yeah, that was a lot of things, yes, but they surround the cell. A phospholipid is made out of a phosphate group and 2 fatty acids, not 3 like a triacylglycerol, but 2. The head is the glycerol, and this is hydrophilic. The tail is the fatty acid, and it's hydrophobic. So a nice diagram would be, like that. So that's the head. That's the tail. Well, both of them are. This is hydrophobic, so it hates water. This is hydrophilic, so it loves water. If you add water to it, then it forms a bilayer. Basically, what I mean by that is when you put in water, the tails hate water, so they try to like minimize the water that gets to them. So even if it's like this, the water will still get to it. So it should have another one over there to protect it. But it still has this side, so it should have one here and one here. This one still has this side, and so on. And that's what forms the cell membrane. If this is the cell membrane, just a portion of it, this is how it looks like. The tails are pointed inward and the heads are pointed outward. That's because the solution outside of cells is mostly made of water. So that's why the phospholipids form a bilayer. The final type of lipid we'll consider is a steroid, and it contains 4 fused rings. So that's basically the definition of a steroid. So that's 1 ring, 2 ring, 3 ring, 4 ring. It looks much more complicated than that, but for this introductory to steroids, yes. An important steroid is cholesterol, and it's the most important molecule in this category. So it synthesizes other steroids, and it's a component of animal cell membranes. So sort of like phospholipids, except it's just like an addition to it. It's mostly phospholipids. And it also creates other steroids, so that's nice too. Proteins. So protein is the most diverse macromolecule. It has a variety of functions. The monomer is the amino acid. It contains amino group, carboxyl group, and side group. So we talked about amino groups. We talked about carboxyl group. What's a side group though? Well, if we have this central carbon, we have an amino group here. We have a phosphate group here. We have a side group abbreviated by R. So this side group is what makes each amino acid different. This all is the same for an amino acid, but the side group makes it different. There are 20 amino acids, and here they are. The non-polar ones are glycine, G, alanine, A, valine, V, leucine, L, isoleucine, I, methionine, M, phenylalanine, F, tryptophan, W, proline, P. So other than tryptophan, which is W, it seems like a nice pattern, right? Wait till you get to the others. They're going to start running out of alphabets so soon that it's going to be like tryptophan. It'll literally be T to W. The polar ones, serine is S, threonine is T, cysteine is C, tyrosine is Y, odd, right? Asparagine is N, and glutamine is Q. And then the charged ones. So non-polar, we talked about, it doesn't dissolve in water. Polar dissolves in water. Charged is basically like an ion. There's aspartic acid, which is D, glutamic acid, which is E. Yeah, I don't know who names these. Lysine is K, arginine is R, and histidine is H. So a polypeptide is a polymer of amino acid, and this creates proteins. Now, notice a protein isn't exactly a polypeptide because, as we'll see later, it's more complicated than that. Peptide bonds are the bonds that connect amino acids into polypeptides, and they're covalent. A dipeptide is made up of 2 amino acids. Polypeptide, like we said, many amino acids, dipeptide 2. I mentioned before, polypeptides are not the same as proteins, and I'll show you why in a minute. But first, a globular protein is roughly spherical, while a fibrous protein is like long fibers. These are very different types of proteins, very different functions, and very different shape, obviously. Some important functions of proteins: enzymatic proteins or enzymes speed up chemical reactions, defensive, protect against disease, storage of amino acids, transport of substances, hormonal, coordination of organisms' activities, receptor, they respond to a chemical stimuli. So we talked about stimuli before. It's basically an external factor that causes some sort of response. Contractile and motor proteins cause movement, and contractile are mostly near muscles. Motor are help you move, like actually move, like walk. And structural are for support. The structure of proteins, we talked about the function, now it's the structure. Primary structure is the linear chain of amino acids. Secondary structure is the coils and folds created by hydrogen bonds. So the primary structure would be something like this. And that's not a protein. It's literally one chain. That's not complicated enough. Biology is not like that. Secondary structure, coils and folds created by hydrogen bonds. Well, if this one was attracted to this one, then for some reason, it'll go like this. And that'll be a coil or a fold. And if that was a hydrogen bond, then that'll form the secondary structure. A helix is held together by hydrogen bonds between every 4th amino acid. So by the name suggests, it sort of looks like a helix. A B-pleated sheet is 2 or more strands of polypeptide chains bonded by hydrogen bonds. So these both are examples of coils and folds in secondary structure. Tertiary structure is the polypeptide shape. Basically, tertiary structure creates the polypeptide. And it's basically interaction between the R groups of the amino acids. So if we had this before, yeah, I know this is just a rough drawing. Then if we, for example, this one is attracted to this because of the R group, it'll form some sort of polypeptide. Yes, exactly. That's a polypeptide. I made it myself. Hydrophobic interaction is basically non-polar R groups are clustered in the core. And it's basically so that it's away from water. And it holds them together because of van der Waals interaction, which we talked about earlier. It's basically how a gecko climbs on a wall. It's weak interactions. Now, we got to tertiary structure, which is the polypeptide shape. But remember I said a protein is not the same as a polypeptide. So there must be one more step. Quaternary structure. It's the overall shape of protein formed by aggregation of polypeptide subunits. I know it's really complicated, but it's basically the polypeptides coil and change shape to form the protein. Now, notice a protein can have more than one polypeptide. So if this amazing was this what I drew earlier? Yes. Polypeptide was one polypeptide, then this could be attracted to another polypeptide that looks something like this, which is attracted to another polypeptide, somewhat shorter, I guess. And I don't know, I'll just draw a bunch of bonds. and then this coils to form a protein. Quaternary structure may include a non-polypeptide component too. It's not, a protein is not just made up of polypeptides. Proteins are much more complicated than that. Denaturation is when the protein unravels and loses the original structure and function. So we don't want this to happen because then protein's dead. We can't do any of the functions, the amazing functions we listed on the previous slide. This can be caused by changes in pH, salt concentration, temperature, etc. Chaperonins, as the name suggests, it's like a chaperone. It basically helps other proteins fold. And it's sort of like a protection barrier when another protein is being made, and it protects it from bad influences, basically changes in pH, concentration, or temperature, anything that could denature it when it's building. Nucleic acids, finally. So a bit of background. A gene is a unit of inheritance, and it has the instructions to put together amino acids to form proteins. That's how proteins are made. A nucleic acid stores, transmits, and expresses hereditary information. So basically forms the genes. The two main types are DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. The DNA is the genetic material that is inherited from parents, and it has the instructions. DNA makes up genes. It provides all the instructions, including DNA division. So imagine that DNA has instructions to split itself in half. It requires proteins to implement the instructions, except for building the proteins. That's the one that is kind of weird, because if it needs proteins to implement the instructions, how can it implement the instructions to build proteins? That we'll talk about when we talk about DNA synthesis, DNA synthesis, DNA division, RNAs, etc. So RNA has different types. mRNA or messenger RNA is synthesized by DNA nucleus and helps the ribosomes synthesize proteins. So we know ribosomes create proteins, and we know DNA has the instructions to create proteins. So how does the DNA instructions get to the ribosomes? That's because of mRNA. The monomer of a nucleic acid is nucleotide, and it's made up of a phosphate group, a nitrogenous base, and a 5-carbon sugar. There are different types of bases. A pyrimidine has one 6-membered ring, and these are called cytosine and thymine, and uracil is only in RNA. Cytosine and thymine are in DNA. A purine has a 6-membered ring fused to a 5-membered ring, and this is adenine and guanine, A and G. DNA has a sugar called deoxyribose, and RNA has a sugar called ribose. Polynucleotides are polymers of nucleotides bonded by phosphodiester linkages, another type of linkage. The 5' end of a polynucleotide is when the phosphate is attached to the 5-carbon, and the 3' end is when it's attached to the 3-carbon. So if this was one nucleotide, the polynucleotide would look something like this. And they connect by the phosphate group. These are the phosphate groups. This is the 5' end because it's attached to the 5' carbon. This is the 3' end because it's attached to the 3' of the carbon. 3' just basically means the 3rd position. 5' means the 5th. So DNA specifically has 2 polynucleotide strands that form a double helix, and it looks something like this, as you've probably seen it earlier in your life. And each of these is a strand. DNA is antiparallel, which means it runs in opposite directions. One strand runs in 5' to 3', and the other runs in 3' to 5'. RNA has one polynucleotide strand and has variable shapes. So some of them is just one strand, like DNA. Some of them well, DNA has two, but it's similar shape, sort of like a one helix, not a double helix, single helix, whatever. And some of them look completely weird, and that we'll talk about later, such as tRNA, rRNA. Those do not look at all like DNA. So yes, tRNA. It brings the amino acids to the ribosome when it's time to create the proteins, and it's called transfer RNA. This is one that looks completely weird. So complementary base pairing in polynucleotides, A pairs with T in DNA, A pairs with U in RNA, and G pairs with C anywhere. So if you have an A on one strand, you have to have a T on the other. If you have an A, well, in DNA. In RNA, if you have an A, you have to have a U. If you have a G, you have to have a C in both. So that's it for our macromolecules lesson. And basically, we just finished introduction to biology. We finished all the chemistry, etc. Now we're going to be talking about cells. The next few lessons would be focused on cells, which is basically the first actual unit of biology. The previous ones were just introductions. So I'll see you next time. Bye.